This is Computer Science and Engineering 428, the Computational Biology capstone. The goal of this course is to get students engaged in design projects that involve use of computers to tackle biological problems. Biology these days is a very data-rich field. It requires computational aids to process this data.
So our project is allele-specific expression, and we're correcting bias and then analyzing before and after we correct that bias how the allele-specific expression changed. So you have two chromosomes, one from your mom and one from your dad, and our tool is trying to analyze what % you express each of those chromosomes.
I'm working on figuring out this tool that RTA created that will generate graphs that show the relative frequencies of each nucleotide based on the start position of a read.
To me, I think there's a huge amount of data, biological data, that needs to be processed somehow, and I think contributing to that effort is important. That, and also I'm just straight up interested in biology.
This course is it's a lot of fun because it gives you a different perspective than just straight up computer science, because you're applying it to a completely different field. There's not very many people who know how important computer science is in biology, and so being able to do stuff like this and just explain to both biologists and computer scientists what I'm doing, it seems like connecting the gap seems like a really cool thing to do.
So one of the major drawbacks of the simple Poisson model is that it doesn't take into consideration at all of the dropouts. The project that we're working on this quarter is for single-cell RNA sequencing. Essentially, the problem is during conventional RNA sequencing process, a number of things could happen that could result in certain parts of an RNA to be dropped out. And what we're trying to do is to try to figure out what part of the dropouts was due to biological reasons and what part of the dropouts was due to technical reasons that happened during the sequencing process.
So with this RNA sequencing project, we're not really trying to sequence the RNA and get it out of the cells. What we're doing is we're taking the data that comes from different cells that you need for different medical analysis, and we're trying to figure out, well, when is the data actually accurate and when is the data not accurate? And when it's not accurate, we're trying to get rid of that information so that we can just focus in on the data that we want so that we can make good medical diagnoses in the future.
I like that, when we meet up, we can talk about big picture problems. Every person sort of finds their own kind of type of work that they want to do, and we do a good job of dividing up, people who want to do parsing versus modeling versus more statistical stuff. So I think we do a good job of getting together, talking big picture, and then dividing up and separating out the work.
I like the kind of collaborative nature of it. I love getting up with the other guys and whiteboarding stuff out and figuring out problems and detailed with the nitty-gritty issues that we have to figure out along the way and then overcoming them.